ਅੱਜ ਕਰਨੀ ਹੈ ਹਰ ਹਾਲ ਚ ਟਿਕਟ ਬੁੱਕ ਸੌਣ ਤੋਂ ਪਹਿਲਾਂ ਕਾਲ ਕਰ ਲਾਂਗੇ ਸਰ ਨੂੰ। ਮੇਰੀ ਕਲਾਸ ਸਟਾਰਟ ਹੈ, ਕੁਛ ਬੋਲਿਓ ਨਾ। Hi there, hello everyone, welcome back. So, yet another session continuing the various molecular biology techniques with a virtual experience bit by bit. Let's wait for another 4 minutes. So by 21:05 we start the session, and we will went into the more, like we have discussed more about things at the cellular level. Now we will things discuss at the mouse level, at the model level, mouse level, rat level. How we create transgenic mouse? How do we create all these to find out new results? Yeah, so we will discuss about them. Oh, oh, oh. There are very few students have joined so far. I think 11, huh? Oh, it's 13 now. 14. I think they get lazy to join in. Hello, 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 everyone. Good, good, good. I hope you had eaten your dinner during this time and might be sleepy to go over the bed and have a good sleep. Even after giving this lecture from 9 to 10, I had really good sleep actually. I feel so like, what shall I say? My biggest task, or not biggest, but my main task in my life that I'm here, that to teach, that I had with me to share with everyone, not to keep with myself. Um, I had a really good sleep also. So that's the thing. I really want you all that wherever you will be in upcoming 5 years or 10 years, wherever you will be, do something that which makes you satisfied, which makes you happy, which makes you want to do it again and again. And believe me, don't worry about money. Money will come to you automatically behind. But the things that you love, whether it's painting, dancing, writing, or I don't know, anything uh, that makes you feel happy, that just bring your you that bring the glow on your face, maybe. Yeah, that makes your inner single cells to work over that work part. Each cell is working and concentrated to finish that job. It could be anything. It could be designing of purse, maybe a designer, fashion designer, or lightning, modular design, lightning, architecture. There are many things in the world. Um, so just think, earning money, maybe making a lot of money. Some people like to make a lot of money. Do that then. So do what you feel the best, which makes you happy. And on that note, we come to science. So now we're gonna discuss about study gene functions, the various methods to study gene function. So studying the functions of gene protein, some basic principles and nomenclature. So mutant organisms, cells. So comparisons of mutant and wild type function, which has genetic analysis, screening of DNA library, and has gene inactivation. Yeah. It has clone genes, DNA sequencing, and they have expression in the cultured cells, and database search to identify protein coding sequence, PCR isolation of corresponding genes. Yeah. So We can see here the mutant organism is giving this clone gene and then protein. This could be reversed with the help of database search and gene activation. So there are four types of mainly types of your what do you say studies that we do: in vivo, ex vivo, in vitro, in silico. In vivo is in living organism like bacteria, yeast, monkey, Christmas tree maybe. Ex vivo is an organic, an organ plant like your tissue biopsy. In vitro in reagent tube like in the cell culture, synthetic setup. In silico is more computer based simulation, more from the computer with the help of bioinformatics softwares. And I was telling in the morning, so this bioinformatics is one of the key player at the moment, which will during this COVID time when all the labs were shut down. So many bioinformaticians they came into play key role to identify the COVID structure, to identify the various inhibitors to control it, just based on computer stimulations. But never mind, these are all stimulations, right? They are not exact results that might be happening in the body. So to do so, as a molecular biologist, we need these molecular biologists are really required to finish that job, to make that thing that what you are showing as a simulation, computer-based simulation, in reality is how is it happening. In order to do so, your molecular biology and biochemistry techniques helps to make it proof. So it's not you can say only single man. It's not a single man army. It's we need all team together and work for the hypothesis, the question, the query, the disease, the disorder that we're working for. Then what are the advantages of working with the culture cells over the intact organisms? So we can get more homogeneous than cells in the tissue, can control experimental conditions, can isolate single cells to grow into colony of genetically homogeneous clone cells, and ethically it's a non-controversial. So commonly used cell cultures are derived from bacteria, yeast, and vertebrates. So growth of microorganisms in culture, it has examples like E. coli and the yeast Saccharomyces cerevisiae. Yeah, have rapid growth rate and simple nutritional requirements. Can be grown on semi-solid agar, and mutant strain can be isolated by replica plating. So this is the bacterial colonies and yeast colonies we can see. So replica plating, in this you take the sterile velvet covered stamp like that, you have colonies on them, and then you transfer this over the minimal medium with arginine or without arginine. So missing colonies, the arginine negative requires arginine here, so they will be not present, but arginine mutant colonies will be there. So the one which is mutant, they will be present at that time. So growth of animal cells in culture, which requires rich media including essential amino acids, vitamins, salts, glucose, and serum. And most grow only on special solid surfaces like blood cells, growth in the suspensions. So this is a single mouse cell. This is a colony of human cells, and there's many colonies in the petri dish. This we have discussed in the animal cell culture deeply also. So this primary cell line, how this culture goes like, so we can see that in the human cells versus mouse cells, it shows a different pattern. The human cells, they show the first phase one like this, going in linear up, and then it's straight, right? And then no changes till phase 2. Then in the phase 3 it started to decline and have the ultimately cell senescence. On the other side, in the bacteria or in the mouse cells, initially there is a loss of potential growth that is happening. Then it has a senescence for some period, yeah, after 30 days, and then afterwards started to go up, and then they become immortals. Yeah, that means they will not die anymore. So one of the fibroblast cultured cells here with the differentiation. You can see very nice circle here with the nucleus inside that, and all the cell organelles around it, rough endoplasmic reticulum, and they're shining. Yeah, this is the very nice part. They started to shine like maybe spiritual, renowned personality. But and these are the fibroblasts can be cultured and differentiated, and undifferentiated cells and differentiated cells. So in non-differentiated cells, we can straightforwardly watch with the help of the dye, red dye that we have used, that the cells are not fully developed. But at the other case, in the differentiated cells, your fibroblasts are pretty much developed and soaring like structure and seems pretty good. Now let's talk about generation of transgenic mice and knockout mice. How to generate your transgenic and knockout mice. If there's a loss of function, that means it's a knockout mice, and it could be done with the help of siRNA also, yeah, for the loss of function. Two ways to do it. And gain of function, that means that is a transgenic mice. And there could be also conditional knockout, knock-in, that you remove from only one specific organ or one specific cell line in the mouse, and there is also tissue specific mouse line. And the mutation, what are the types and causes? So organisms, genotype is its entire set of genes and may denote whether an individual carries mutation in one or more genes. So an organism's phenotype is its functions and physical appearance and depends on that individual genotype. So normal organisms may develop changes termed mutations in their DNA sequence, thereby altering their genotype and perhaps their phenotype. So organisms that develop such mutations are termed mutants, and mutations may be part of the genotype or occur as acquired mutations. So an haploid organism has a single copy of each chromosome, and its phenotype is consequence of that one copy. Whereas a diploid organism has two copies of each chromosome and two copies of each gene. So there's homozygous and heterozygous. Just remembering your previous genetics basic knowledge here. And diploids that carry identical alleles and diploids carry different alleles. They are called heterozygous. And the two copies of each gene may be same or copies may be different. So different form of each gene are termed alleles here. So alleles may be dominant or recessive. So diploid genotype, it has a wild type, and the diploid phenotype would be also wild type. Yeah. In the mutant one is dominant is present, the mutant. Another is wild type is present with nothing, and there's also both dominant present. Then it's also mutant. One is recessive, then it's a wild type. Both are recessive, then it's a mutant. So some genotype linked diseases here: sickle cell anemia, cystic fibrosis, phenylketonuria, Tay-Sachs disease. Yeah. So these are the molecular and cellular defects having the various European regions they belong to, and some autosomal dominant disease, Huntington disease, hypercholesteremia, and the X-linked, sex-linked recessive genes, Duchenne muscular dystrophy (DMD), and hemophilia A. Some names to remember. So some examples to see how these are genotype linked diseases are working. So this is autosomal dominant Huntington disease, where we can see the male part having the affected region, affected genes. Huntington disease is present on the allele, and in the female part it's not present. So it has babies as a male and females. So the one which carries the HD part, it will be affected, which doesn't, it will be not affected. Then comes the autosomal recessive cystic fibrosis. It will has carriers, both will have the carrier, right? Then it will have the CFTR and it will also have the CFTR. Both male and female, they have the carrier of the your cystic fibrosis gene. Then in the male and females, then further what will happen? The one which carrying the both CFTR will be affected, but the rest will be only carrier, and this will be a non-carrier. On the other hand, the X-linked recessive, this is DMD, this is Duchenne muscular dystrophy, where your male is X plus Y, where female is your carrier with the DMD gene. So the male is affected here because it has the on the X genes, X DMD is present, where others remain unaffected. This will remain again carrier, but the female become non-carrier here. So it's all depending upon the X-linked, when if it is present on the X-link, if it is coming with the male, it's become affected. If both CFTR genes are present, then it's become affected. If only one is present, then it's affected. Yeah, so different conditions, different results. For example, mutants in Drosophila, we can see this red and white eye. That's the basic model that we use in genetics. So, and also here we see normal with the two wings, and in the UBX mutant it has four wings. Yeah. So mutations may cause only subtle changes or may produce significant changes in development, cellular function, and appearance or behavior of the gene. So gene replacement and transgenic animals. So it is possible to replace an organism wild type gene with an active gene to create a gene knockout. So it is also possible to introduce additional genes to create transgenic organisms. And gene knockout, gene and transgenic techniques can involve mutagenesis of clone genes prior to transfer into the organism. That is your in vitro mutagenesis. So mutant organisms, the comparisons of mutants and wild type functions, and it has clone genes DNA sequencing. So genetic analysis, it includes the screening of DNA library, and if it is go back, then gene inactivation that we have discussed at the start of our lecture. Now we have discussed some basics behind this transgenic mice. Uh, yeah, you're welcome. You're welcome. This was the very basics behind it so far. That how majorly your genetics, your basics of your transgenic mouse line works. Now let's see how they work in practically when you want to work them with them. This is one of the interesting part actually. Literally, if you're in research, somebody asks you to create a knock-in mice. It's actually one of the difficult tasks. They are big companies like Charles River. They do this on the like 5 months contract. They give you this knockout mouse after 6 months, and you can grow as many as you want later on. It will give you a lot of good papers, yeah, for the publication. But beyond that, yeah, it's a it's a revolutionary field actually to create this knockout mouse. So let's see in the presentation mode first, then we will see a video mode about that. Yeah, so we will do both things together. So in this first, what you do, just listen to me here now. You inject your embryonic stem cells. Yeah, you take embryonic stem cells into the blastocells cavity of the early embryos. So you have a brown mouse with the gene X minus X plus AA, and the black mouse gene with the AA X plus X plus, which is having 4.5-day blastocyst, right? And then you surgically transfer embryos in pseudo pregnant females. Yeah, then you surgically transfer embryos into pseudo pregnant female. So once this embryonic stem cells they go into the foster mother, then the possibly progeny, because you have a mixture of that, one would be chimeric and one would be black. Chimeric means it will have mixture of brown lines over it and with the gray green lines and brown color, and one will be totally black. Then you select this chimeric again, and then you cross with the wild type black mice. You cross it with the same. Then what you will have, you cross them together. So this will have the all germ cells AX plus, this will have these genes AX minus, AX plus, AX plus. Then ESL derived progeny will be brown. So you will have this progeny from ESL derived germ cells. So the chimeric part will be removed from it, and this black will be also produced out of it. But our interest is only in these genes. Then progeny from these embryonic stem cells derived from germ cells, we screen for the brown progeny, that is X minus X plus, and then mate with the X minus X plus, then screen progeny DNA to identify the X minus homozygotes. Then we see a knockout mouse afterwards. So in this way, we can remove the X minus. That was our task to do so. So initially in the brown mice, it was X minus X plus and an X plus and X plus. So we wanted that it should be X minus. In order to do so, we need to do multiple crossing with each other, and having these progeny with X plus and X minus, we keep screening, we keep mating with the X minus and X plus until we get X minus, so that the gene that we wanted to remove out of our species, it could be removed out. So that is known as knockout mouse. So there are some genes like NO, nitrogenous oxidative NO genes, or they are PP1 protein, or they are PKA protein. There are many proteins, kinases are present, many phosphatases are present, many GPCRs are present in the cells, and we want to check, let's say P2Y2 genes we want to remove out of our mouse cell line. So we will use this technique in order to remove that. Then we will start checking the then results, that how is the lung is, how is the liver is, and and over this, after removing that gene, we will do some different experiments. Like we will make one mice, we will add some cancerous cells into it, we will create a tumor, and then we will check whether the knockout mouse is able to recover off the tumor or not as compared to the normal one. So in this way, you can do many experiments, you can check many physiological functions, and in this way, your drug discovery, your all big pharmaceutical companies that are producing these drugs, things out work out. So that's why these molecular biologists or biochemists, these people, they're the key role. They're working at the very basic, at the very starting point of the making one medicine in order to recover one disease. So we are also, as a molecular biologist, playing a big role in order to heal the universe. So cell type specific gene knockouts in mice. So we have this further LOXP mouse and Cree mouse, and it's getting more complicated now. You should not be worried about these things actually. Uh, Nowadays, in the most, if your lab is rich, so there is also a thing called lab is rich, lab is poor. We should not say the lab is poor. It's very bad to say that word. Sorry for that. Sometimes funding is a problem. At the end of the day, your research is going on. There is also big thing to have fundings, to have big money from your research, because you can keep sitting, keep reading papers, but if you can't bring out the money out of it, and it have no use for the world. So you need to be very passionate about science, be competitive in the society, and show that you can bring out something that is no one has created before. And it's one of the revolutionary thing that no one has done that. That should be the key. So LOXP mouse, all cells carry endogenous genes with LOXP sites flanking exon, and heterozygous for gene X knockout, all cells carry CRE genes, CRE gene. So you have LOXP site, and this is cell type specific promoter. So cells not expressing the CRE in this case, so gene function is normal. So LOXP CRE mouse, they are the cells which carry the LOXP modified gene X and one copy of gene X knockout and CRE genes. So that's how they make it. So cell expressing CRE. So this is how 1, 2, your 3 genes are there, and here we have a CRE protein, and we remove this second part from out of it, and then the gene function is disrupted in this way. There is another way. This is siRNA, which is most famous technology being used. I also use many times in my experiments, and very successful. You do double knockout with the help of siRNA. You remove your things working at mRNA level. Yeah, you stop making mRNA so that your protein will be not produced. Then you can check the functions that when the protein is not being produced of the specific mRNA, so what will be the phenotype of your cell line. So in vitro production of double-stranded RNA, which has a sense transcript and antisense transcript, and then dicer is there, then the siRNA is there. Then you non-injected and injected, and siRNA designed to base pair with specific mRNA, which will then be degraded. So dicer is an endogenous enzyme which cleaves specific RNA structures, and there's in vivo production of double-stranded RNA. Here we can see hairpin construct, shRNA, and we have dicer and siRNA. So in this way, we can create a new species also. Then production of transgenic mouse to increase one gene, specific gene. So we were talking about so far knockout mouse, siRNA technique. Now third part is your transgenic mouse, in order to increase one functional part of the gene. So first is you inject your foreign gene into one of the pronuclei, then fertilize mouse egg prior to fusion of male and female pronuclei. Then transfer of injected eggs into foster mother, and about 10 to 30% of offsprings contain injected foreign DNA, and foreign DNA is present in equal amounts in all tissues. Yeah? Then mice expressing foreign DNA are bred to continue, and DNA in the germline further. So the foreign DNA which contains your GOI site and promoter. If the promoter is a constructive active promoter, then the GOI will be expressed. If the promoter is regulated by protein only present in cells, a tissue specific expressions can be achieved. So you have a transgenic mice, GH plus, like let's say the growth hormone. So we can see in this mice, when we added this growth hormone into the mice with the help of this plasmid vector PBR322 DNA, most highly used plasmid vector. So mouse are bigger, and these are smalls. So this technique, even in the this Khali who went into the WWE to USA, he has also this transgenic problem. He has a lot of growth hormone. So all the people you see tall, big than the normal, they all tend to have lots of growth hormone in the body, which makes them like that. Yes, the same kind of RNA. You're right, you're right. So these techniques could be used in plants and animals and anywhere that you want to use for. They are universal. Now I will dedicate your next 30 minutes over the videos. If video starts to lag, don't worry. I will also provide, like in the evening, I provided you all the videos that we had. So I will also provide these two videos later on. Yeah. So we can start now. I will not speak because of the, yeah. This is CRISPR-Cas9, students. How we create
Gene knockout is a common technique in studying gene function. In this practice, the gene of interest is disrupted by altering the cell's genomic DNA. Any phenotype that results from gene knockout gives insight into the function of the disabled gene. Conventional knockout experiments are frequently achieved in animals, but need a huge investment of time, money, and labor. Therefore, these experiments are usually restricted to well-funded labs or core facilities. This has presented a large hurdle for researchers exploring gene function. The breakthrough of CRISPR technology has turned the tide and made gene knockout a much more accessible tool for many more research labs. The key revolution brought about by CRISPR is the creation of double-stranded breaks sequenced specifically. With CRISPR, scientists can create double-stranded breaks at virtually any site in the genome. To repair the double-strand breaks generated by CRISPR, there are two main mechanisms: non-homologous end joining and homology-directed repair, also known as homologous recombination. With non-homologous end joining, the ends of the double-strand breaks are simply reconnected together, and this process is highly error-prone, introducing mutations, insertions, and deletions, and in many cases resulting in gene knockout. Homology-directed repair, on the other hand, utilizes a repair template, where desired changes are flanked by left and right homologous sequences. Through homologous recombination, the desired changes are incorporated into the genome. The CRISPR knockout knock-in kits utilizes the homology-directed repair system. To plan an HDR-backed knockout experiment using plasmids, the researcher needs to create the following constructs. 1. A plasmid coding for the gRNA, targeting the intended site for double-stranded cleavage. 2. A plasmid coding for the Cas9 protein. And 3. A plasmid serving as the repair template, with a designed sequence flanked by the homologous arm region of the cleavage site, or donor vector. Origin has created pre-designed kits to make knockout experiments even easier. For each gene locus, we have created a specific kit. The first two elements are provided in a single all-in-one vector. The gRNA vector, targeting the start of the coding sequence, was cloned into a vector containing the expression cassette for Cas9 protein. The donor vector was also created with a knock-in GFP puromycin cassette, flanked by the homologous arms of the chosen cleavage site. This permits the selection of knockout cells via puromycin marker. The promoterless GFP provides additional utility to monitor any upstream promoter activity that might be present. To start your knockout experiment, simply co-transfect a gRNA vector with a donor DNA plasmid into the cells. The Cas9 enzyme will cut a double-stranded break near the start codon, and through homologous recombination, the functional cassette will be integrated into the genome, replacing target gene. In the end, 3 events are achieved. 1. The target gene is knocked out through gene disruption. 2. GFP will be driven by the endogenous gene promoter, providing added information of promoter strength. And 3. The puromycin marker under the PGK promoter is integrated into the genome, enabling the selection of successfully edited cells. The protocol starts with co-transfection. There will be 3 separate experiments, one of the 2 gRNA vectors co-transfected with the donor vector, scramble control with donor vector. After transfection, the cells must be passaged for about 20 days before puromycin selection, as the donor vector alone before genomic integration also provides puromycin resistance. Culturing the transfected cells for 20 days ensures that the episomal form of donor vector is lost. This diagram shows the details of transfected cell passaging prior to puromycin application and selection. For HEK 392 cells, 48 hours post-transfection is considered G1 cells, at which point the cells are split 1 to 10 and grown for 3 more days. At G2, CRISPR-targeted cleavage and gene editing should be finished, but the percentage of edited cells will be small. You can perform optional genomic DNA analysis using G2 cells. If the data are negative, you can redo the analysis after puromycin selection. After G7, apply puromycin selection using the doses appropriate for your cells. To summarize the CRISPR knockout knock-in protocol described so far, Step 1. Co-transfect one of the gRNA vectors with the donor vector, along with the appropriate controls. Step 2. Passage transfected cells for around 20 days. Step 3. Apply puromycin selection and isolate individual cell colonies. Please note that the drug selection doses need to be determined by kill curve for each cell line. 5 days after puromycin selection of transfected HEK293 cells, almost all cells in the 2 control transfections died. However, as expected, the co-transfection with the gRNA construct produced puromycin-resistant cells. The 4th step is to analyze the puromycin-positive cells to detect the knockout. If you have a good protein-specific antibody, you can use Western blotting to evaluate the knockout. It is best to use this method after isolating single colonies. Another method is genomic PCR to verify the correct GFP puromycin integration in the genome, followed by sequencing to confirm the integration. To amplify only the edited allele, design PCR primers following these rules. The forward primer should be upstream of the 5' end of the left homologous arm of the donor vector, and the reverse primer should be in the GFP region. In this example, knockout ATG5 gene was conducted by the protocol. Genomic PCR products of the correct size were amplified from the gRNA constructs, plus the donor co-transfected cells, and not from the scramble control transfected cells. The PCR products were further sequenced using the PCR primers to confirm the correct integration. Sequencing data using the forward primer shows the correct integration of the 5' end of the left homologous arm in the donor vector. The reverse sequencing primer confirmed the correct integration of GFP puromycin cassette. The original ATG5 gene was replaced with GFP. Origene offers genome-wide human and mouse CRISPR knockout knock-in kits. With these complete, ready-to-use kits can knock out almost any gene from the genome and knock in the GFP and puromycin cassette simultaneously. You can study the consequences of the gene disruption and potentially do a promoter study at the same time. Origene also has other functional cassettes to provide different fluorescent proteins, luciferase, or different selection markers. If you wish to have the CRISPR knockout kit created a different donor vector, please visit our website to request a custom project. To learn more, please visit the Origene website and our YouTube channel.
Okay, so this was all about students, participants here. CRISPR-Cas9, yeah, will be very, in 2016, I don't remember the person, she got the Nobel Prize winner for this CRISPR-Cas9, and it has saved a lot of time, and it has saved a lot of money, and it have created a really revolutionized to create a new mouse line, to create everything in just a few like days. So which was earlier like so many days we were using adenovirus, siRNA, and so many things. CRISPR is the thing. CRISPR is the future. So that's one of the best technique. Just go through. I hope you had a little bit understanding with having this Cas9 and everything, cutting it and then making a new genetic material. But it will be it's a 2020 chemistry Nobel Prize. Okay, I was not aware. Yeah. Okay. Jennifer Doudna, Emmanuel Charpentier were the scientists, I guess. Okay, great. Thank you very much for this information. Thank you, Nidip. Thank you, Akshay. Yeah. So, and you know, there is also call on LinkedIn in order to be motivated, because there are some dark clouds over us. Even I had some days, we have very foggy day, that on your brain is everything foggy. You don't know what to do, what's going on in life, or how things are happening. I don't know what will happen in future. how things will prevail in upcoming time. So I usually just this makes me, during this COVID time when everything was so this one everything was so, like, dead, and it was so horrible time that we have seen this 2 times. I just tend to go on over this page. It has so much to, like, if you're passionate about science, if you want to go something big, and you don't want to settle for small, you want to settle for big. Yeah, because if you think big, big will come. If you think small, small will come. Yes, it's always like that. The bigger the vision, the bigger will be the outcome. The smaller the vision, smaller will be the outcome. So these pictures, stories, the old pics. Earlier, when I used to see these pics in our books, I was not mature, to be honest, at that time in my college days. I like, okay, they did something. But now, when I work, I went through this life of, like, um, 7 days a week in the lab, working from morning till evening, even going in late night to check your cell line if it is fine. And then you see people who are very successful, and they did the same thing that we were also had done ourselves. It's very remarkable. Nelson Mandela, wow, he is one of the, yeah, sentenced in the year 5 years in prison. No, stayed 27 years. Sorry, it's from South Africa. Very highly remarkable people. Carol Greider, she got the Nobel Prize in 2009. At just age of 23, she got her PhD. Can you believe that? She found the telomerase. On the chromosome there is a cap, telomerase. So she found that telomerase enzyme that creates that telomere over the chromosome. She's into the water, finding some organisms in it. So it's just funny and very, like, motivated thing. So yeah, so I just keep sharing things which keeps you motivated to go on, to go further, and not to stop. Yeah, that's why I said, students, you can send me also connection request and follow this page also. I will be happy to be joined with everyone. Okay, now, so we have, these 3 days we have discussed the basics of molecular biology, animal cell culture, microscopy. Today we have discussed so many techniques about molecular biology. Now I want to take you to the industrial perspective, from the fermenter perspective, how the real things work in the industry, how an industry thing work. So there will be a bit 20 minutes video about it, which will be explaining very core details how, and it will tell how, what is the meaning of aseptic. Everyone is wearing a lab coat, everyone is covered by themselves, so neat and clean. There is no, on the instruments, all the machines that they're using, no rust is there, everything is superb and stupendously. I really admire the working that is happening in that lab, in that industry. So I'm gonna start that now, so enjoy this late evening industrial applied molecular biology, applied industrial application.
The ability to insert or knock in a gene at a specific
Okay,
locus is a
is this one? No. Okay, so I will play. Sorry, that will be, I think, some other day. But this is just to explain knock in and knock out. Just 5 minutes video.
The ability to insert or knock in a gene at a specific locus is a tremendously useful method for dissecting disease mechanisms. However, in previous years, the insertion of a foreign gene has been far too tedious and expensive for many labs to perform. Thanks to the recent development of CRISPR-Cas9 technology, gene knock-in experiments have now become a very popular technique in modern molecular biology. In contrast to the random integration events by the lentiviral system, CRISPR utilizes a guide RNA that directs gene insertion with extraordinary specificity. In particular, The AAV S1 safe harbor site is often a preferred target site for gene knock-ins. This is because it enables consistent and robust transgene expression with minimal toxic effects. At this safe harbor site, the surrounding DNA maintains an open conformation, enabling stable expression of the newly inserted transgene. This makes safe harbor sites, such as the AAV S1 locus on human chromosome 19 and the ROSA 26 locus on mouse chromosome 6, very popular sites for targeted knock-in experiments. In this case study, we will walk you through how a red fluorescent protein gene was knocked into human embryonic kidney cells at the AAV S1 safe harbor site. We will also demonstrate how the knock-in was verified using a PCR-based method. To begin, an sgRNA was designed to target the human AAV S1 locus. Software analysis was performed to ensure maximum efficacy of on-target cleavage while avoiding any off-target effects. The resulting guide sequence was used to construct ABM's AAV S1 targeting all-in-one vector. This vector expresses the Cas9 endonuclease under a CMV promoter and the sgRNA under a U6 promoter. To achieve the knock-in, a donor plasmid containing the gene of interest is required to provide a template for homology-directed repair. The HDR pathway is a mechanism in cells that repairs double-stranded breaks by using a DNA repair template homologous to the sites flanking upstream and downstream of the break site. To enable HDR-mediated gene insertion, the gene of interest must be flanked by such homology arms. Therefore, a second plasmid was constructed by cloning an RFP pyromycin expression cassette into ABM's repair template plasmid for the AAV S1 locus. The resulting AAV S1 donor RFP plasmid had the RFP gene under a CMV promoter and a pyromycin resistance marker under a PGK promoter. Once both plasmids were ready, they were introduced into HEC293 cells using ABM's transfection reagent, DNA fectin. Following transfection, the cells were passaged 10 times to dilute out the episomal donor vector. Following, the resulting HEC293 cells were given 3 to 4 weeks to grow in the presence of pyromycin to select for the cells with successful knock-in of the RFP pyromycin resistance cassette. Cells transfected with both AAV-S1 targeting all-in-one vector and the AAV-S1 donor RFP plasmid were healthy after 4 weeks, and greater than 95% of HEC293 cells were successfully expressing RFP. Control cells that were not transfected with the vectors died after pyromycin treatment. PCR genotyping was carried out to confirm that RFP was successfully knocked in at the AAV-S1 site. In this method, 2 primers were designed. Primer 1, targeting the 5' homology arm upstream of RFP, and Primer 2, targeting a site within the RFP pyromycin resistance cassette. Gene knock-in was confirmed when a PCR product of 1.1 kb was observed, indicating that primers 1 and 2 were both able to bind to the edited genome to enable successful PCR amplification. In the case of a non-successful knock-in or in the presence of wild-type control cells, no PCR products were observed, as primer 2 could not anneal to the target site and enable amplification. We hope this case study has demonstrated how the CRISPR-based gene knock-in technique can make your exploration of gene function amazingly easy and efficient. CRISPR-Cas9-mediated gene knock-in allows for biallelic modifications of some genes to be generated with ease, with phenotypic changes in mice observable in a single generation, an unprecedented time frame. Let ABM's team of CRISPR scientists help you knock in any gene into any cell line you want, all with minimal toxic effects at the AAV S1 or ROSA 26 safe harbor sites. Explore ABM's extensive portfolio of customizable CRISPR services and products today, and start knocking in genes, tags, markers, and more. To make it even easier for you, simply describe your desired knock-in, and we'll take care of the rest.
So that's it for today, student. I will not make things more more things. That the one I was saying that about all that, that will be day fifth actually. After we do the proteomics part, all the 2D gel electrophoresis, then that will come because that will make sense on that day, not today. So today, that's it for today. I don't want to make start. We still have time to the day fourth applications. So where it is, I do notch. No, I do fabrica. MBBT day fourth. Yeah, day three is finished. So I have shared everything with you. Maybe some videos need to be shared. That one, the last two videos with the CRISPR Cas9. I will share them now. This one, right? This one. Okay, then take care. You're good to go. Sleep well. Good night. Ciao, ciao. Bye bye. Good night. See you tomorrow again. So there will be no holidays over the weekend. We will continue our classes tomorrow as normal, as usual. So we will see us at 5. Yeah. And I will not go tomorrow. I will stay at home then. Okay. Bye bye. Take care.